They look like a donut. All right, and there's a reason for that. Platelets. Platelets are cell fragments. They are not a complete cell. And for the rest of these formed elements, it's going to be our leukocytes. Leuko meaning the white, okay? So these are the white blood cells. They are put into two categories. They are either classed as a granulocyte or an agranulocyte, meaning without granules. At the time these categories were made, the ability to view the cells is not as great as it is now. They all have granules. But in the beginning, we couldn't see all of the granules. So, the cells that do have very visible granules. And can you guys see what I mean by granules? Okay. My neutrophils, eosinophils, and my basophils. Under the scope, when you look at them, when you look at a blood sample under the scope, the red blood cells, very definitive. The white blood cells, very definitive. Platelets, very definitive. It then becomes the issue of trying to determine which of the white blood cell it is. Is it a neutrophil, eosinophil, basophil? Under the agranulos, the lymphocytes, the monocytes. One of the things about the white blood cells, they keep a nucleus. Red blood cells don't. Let me say that again. White blood cells keep the nucleus. And for each one, the nucleus structure is different. It's one of the ways that they can identify the white blood cell in a blood smear. Red blood cells do not keep their nucleus. Did y'all know that? Do you have any questions about that? Why? 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 Huh? Because so far, we've always been taught that that nucleus is the brain of the cell. And now I'm telling you that something very important, a red blood cell, doesn't have it. That's going to be interesting. Like I said, we can separate the components of blood. Anybody in here donate blood? Okay. And that means you know your blood type. Okay. I used to know mine. I can't remember. And now, when you hear, for example, the commercials about donating blood, it tells you you can save 3 lives from your donation. Why? because you might have the same blood type, but that would be based on the red blood cell portion.
Is it because whatever type of blood type you are, you could go to everyone, like your blood condition?
Once again, the red blood cell portion. How many layers? 3. When you donate blood, they now do this. It is centrifuged down because there are 3 layers that settle out. The red blood cells are the heaviest, so they settle out to the bottom. The white blood cells, the buffy coat, they come in to the middle, and you see that that's a very small amount. If you were sick, that buffy coat would be more. And then you see the plasma that contains ions and nutrients and all these other components that are flying around. Because, like, at the time of blood loss, that's the part they need. Okay? Someone who may have an immune deficiency may only need the buffy coat. Someone who may have something wrong with IGFs, other molecules like nutrients and so forth, they may need the plasma portion. You've saved 3 lives with the one giving up blood.
What are their plasma donation centers then? Is that, and they strictly take blood?
They strictly take the plasma because when people donate blood, okay, one of the things that, well, they check now for a ton of stuff, but they have a huge need for the blood and the plasma and so forth. If that person, now get this, you know how, like, if we get sick with a virus, i.e. a cold, okay, we tend to build up the white cells that are going to be available for the next time you might come across that virus. And you don't get sick. They need the people who don't have any of that. And that's a huge one that they look for. They look for in that blood for it to not have certain viral strains in it. And because that would be part of it, they simply take the plasma so that they can, for the most part, give it to babies. So if a person goes to give blood, and if they're negative on certain materials being present, they want to only collect that person's plasma. They actually, as the blood is being collected, return the red blood cell portion back to the donor. It's very interesting what they can do now with the collecting of blood. So when we look at what when we're looking at a blood sample from a patient, okay, and certain information that we can gain from it, look at what we can get. Now, about 8% of our body weight is the blood. Like I said, in any given moment, we usually have about 4 to 6 liters. That amount can increase and decrease in the body, and the body can adapt to that for a short period of time. But under normal conditions, about 4 to 6 liters. Temperature, we can get from it. pH, we can get. Viscosity, we can get. We can get osmolarity, which is something that hopefully they covered in your Part 1 course. Is everybody familiar with osmolarity? an osmole, oh boy, you're going to need to know that going into your program. Do you know what it is? Okay. When we talk about osmolarity, because one of the things that will show up on lab work, it will be given to you in milliosmoles, meaning a millionth, okay? Because an osmole is going to be referring to a molecule in the blood. And it's based upon, does it break apart or not? Let me give you an example. What is that? Glucose. What's glucose?
Carbohydrate.
Sugar, carbohydrate. Why is it important?
To make ATP.
I need it to make ATP. It is the simplest sugar that our body can use. Any sugar we have in the body has to be converted to this for our body to utilize it. Does that make sense? It's the only sugar our body can use. Does it break into its component parts in the blood? Does it break it? No. It keeps C6H12O6, the structure. Does that make sense? So now, if I were trying to determine milliosmoles of that, or osmoles, okay, because it does not break apart, it is simply considered 1 osmole. So, if we had that in a liter of solution, because you can see it's given per liters or milliliters, because it becomes a millionth in the body, I would simply have 1 milliosmole per 1 milliliter, because it did not break apart. But what about this? What's that? Sodium chloride. What we tend to know as salt. That's not the whole component of salt. Everybody knows that, right? Because when you take in salt, salt that we eat has sodiums, potassiums, magnesiums, all these other things, okay, that the body needs and might not need, all right? But we talk about sodium chloride because it's kind of easy, okay? Now, if you take this and put it into water, fluid, okay, does it break apart? Emily?
Yeah.
Into what? Na plus, Cl negative. It breaks into ions, and it broke into 2. I now have 2 osmoles.